My name is Shaji Kumar. I'm a consultant in hematology at Mayo Clinic in Rochester. We just concluded the 2020 ASH meeting, which was completely virtual this year because of the COVID pandemic. A lot of exciting data were presented at the meeting, both in terms of understanding the disease biology of multiple myeloma and related disorders, as well as early results from some exciting clinical trials, which in the next few years will change how we manage patients with multiple myeloma. There were quite a few studies related to understanding the disease progression from smoldering multiple myeloma to active myeloma, particularly in terms of looking at genetic changes to predict the risk of progression. There are clearly ongoing trials that are looking at the impact of early intervention in smoldering multiple myeloma. In the area of newly diagnosed multiple myeloma, there were longer-term results of several clinical trials which will impact our current approach to treatment of newly diagnosed disease. One thing that was clearly evident from three large trials was the continuing role of autologous stem cell transplant as part of initial therapy of multiple myeloma. The IFM 2009 trial with almost 8-year follow-up confirms the progression-free survival advantage of doing an early autologous stem cell transplant while still demonstrating no overall survival differences between patients who got a stem cell transplant in the upfront setting versus getting a stem cell transplant in a delayed fashion, suggesting that in selected patients, if they prefer, we can certainly delay an autologous stem cell transplant. The Forte trial also looked at the question of autologous stem cell transplant in the context of a different induction therapy with carfilzomib, lenalidomide, dexamethasone, again showing that use of an autologous stem cell transplant, even when you use a effective regimen like carfilzomib, len-dex, still adds to the outcome with better progression-free survival with the transplant arm compared to the no-transplant arm. The trial also confirmed that a combination of a proteasome inhibitor and an immunomodulatory drug like lenalidomide is the best combination to use in the upfront setting for these transplant eligible patients compared to using an alkylator with a proteasome inhibitor combination like carfilzomib, cyclophosphamide, dexamethasone. Finally, the EMNO2 trial had longer-term follow-up, which clearly demonstrated that patients who got a stem cell transplant as consolidation did much better than the patients who got VMP as consolidation instead, and also went on to show that patients who got VRD consolidation post or autologous stem cell transplant had a better outcome. As you all know, the prior studies had shown, or previous reports of the study had shown that patients with high-risk myeloma appear to benefit from using a tandem autologous stem cell transplant. So the essential takeaway message for the transplant eligible patient population right now would be that they should be getting initial therapy with a proteasome inhibitor and an immunomodulatory drug combination, followed by a single stem cell transplant, and maybe consolidation depending upon what injection therapy you use, but certainly putting them on lenalidomide maintenance post-autologous stem cell transplant for standard risk myeloma. And then for high-risk patients, considering a tandem autologous stem cell transplant and maintenance with a bortezomib or a proteasome inhibitor and lenalidomide combination. Now, in the transplant ineligible patient population, there was again long-term follow-up data from the Maya trial, which looked at daratumumab, lenalidomide, dexamethasone compared to len-dex. With 4-year follow-up, now the PFS benefit continues to be evident with the median PFS not yet reached for the 3-drug combination. The estimate would be that we probably will reach a median of 55 to 60 months with daratumumab len-dex combination used upfront and continue till progression in patients with transplant ineligible multiple myeloma. Now, in addition, there was a larger phase 3 trial that was presented, which is the Tourmaline trial, which looked at the combination of ixazomib with lenalidomide and dexamethasone compared to lenalidomide dexamethasone in a transplant ineligible patient population, fairly similar to what was enrolled in the Maya study. And that trial again showed that there was approximately a 12-month improvement in the progression-free survival with the use of the 3-drug combination compared to the 2 drugs. However, the trial did not meet the pre-specified threshold for improvement in the PFS. Now, both these, all these trials have looked at the concept of using a 3-drug combination in the setting of transplant and non-transplant eligible patients for newly diagnosed myeloma. Now, there were also data from trials that are looking at quadruplets for initial therapy of multiple myeloma. As you all know well, that Alcyone trial had previously had shown that adding daratumumab to VMP improved the outcome of transplant ineligible patients as newly diagnosed treatment. We also have previously reported data from the Cassiopeia trial showing that addition of daratumumab to VTD pre-transplant as injection therapy, and again, use of daratumumab versus observation in a randomized fashion post-transplant. This approach certainly led to an improved PFS for the four-drug combination. At the ASH meeting, there was updated results presented from the Griffin trial, which randomized patients to VRD versus daratumumab VRD before stem cell transplant, as well as 2 cycles of consolidation post-transplant. And then patients continued on maintenance with their respective regimens, either DARA, LEN, or LEN. We still don't have a PFS improvement data from this clinical trial yet, but we know that the depth of response was significantly better, especially the MRD negativity rate with the 4-drug regimen compared to the 3-drug regimen. So obviously we had to wait and see what the survival data would show before the quadruplets would be routinely incorporated into practice. Another area that needs to be highlighted is the role of minimal residual disease testing. There were a number of abstracts, both from correlative studies done as part of clinical trials and otherwise, that demonstrated the importance of minimal residual disease testing. One of the concepts that have come up in the past is whether we can limit the transplant to patients who are MRD positive, or use maintenance only in patients who are MRD positive. And the longer term data from both the IFM 2009 trial as well as other trials have shown that it's still too early to say that patients who are MRD negative does not benefit from these interventions. So we certainly need more data to see where we can use MRD testing as a clinically useful tool or clinically actionable tool. Now, finally, another update on the maintenance setting would be the data from the Forte trial that used the carfilzomib plus lenalidomide maintenance, comparing it to lenalidomide maintenance. And they have shown that there's a PFS advantage to KR maintenance followed versus len maintenance. However, we certainly want to wait and see what the longer term follow-up would be for these patients as well. So certainly, I think one strong takeaway message from the ASH is what the initial therapy should be for the newly diagnosed myeloma. What is the role of transplant? What is the role of maintenance post-autologous stem cell transplant? Now, in the setting of the early relapse, and this is an area that is of great importance because the majority of the patients who actually suffer their first relapse are often on continued maintenance with lenalidomide or have received significant amount of lenalidomide for a considerable amount of time and are often not very sensitive to this drug at the first relapse. So in those patients, the question always comes up, do we use a pomalidomide-based combination or do we use a PI like carfilzomib-based combination? So there were updates from the IKIMA trial that used isatuximab in combination with carfilzomib, as well as Candor trial that looked at daratumumab in combination with carfilzomib. Both these trials, even with longer term follow-up, shows that the median PFS after at the time of the first two relapse, or the first few relapses, is roughly about 26 to 30 months. There's also data presented from the Apollo trial that looked at the daratumumab in combination with pomalidomide dexamethasone, showing a 12.5-month PFS compared to 9-month PFS with PomDex alone. And there was also updates from the ICARIA trial that looked at isatuximab in combination with pomalidomide dexamethasone versus PomDex. So when you put the data from all these 4 trials together, the key message is that when patients relapse on len maintenance, you have a choice of using either one of these monoclonal antibodies in combination with either carfilzomib or pomalidomide. And depending upon how refractory they are to lenalidomide, you may want to use a PI-based combination, or if they are not refractory to lenalidomide, or if they prefer to use an oral drug, then use a pomalidomide-based combination at that point. Now, finally, I think there were a number of presentations related to use of immunotherapy approaches, particularly the CAR T cells as well as the bispecific antibodies. In addition, there was also data presented on a new antibody drug conjugate. Now, from the CAR T perspective, we saw about 4 or 5 different CAR T cells that were presented targeting the BCMA or the B cell maturation antigen. Most of these trials enrolled patients with a median of 5 to 6 prior lines of therapy and had response rates of about 70 to 100%. And again, we don't have longer term data except for the BB2121, which showed that the median PFS is roughly about 8 months, even with the longer follow up. But the overall survival appeared to be almost 3 years for this group of patients enrolled on that particular trial. Now, one of the problems with the autologous stem cell, the autologous CAR T is the time it takes for creation or manufacturing. And so there is clearly a role for something that can be done off the shelf. So there was data presented with the um, allogeneic CAR T cell that also very early data, but shows that you can get responses of about 50 to 70% in heavily pretreated patients without a significant risk of graft versus host disease in those patients, and a median of about 5 days from the enrollment in the study to treatment. Now, in addition, there was also different approaches to creating autologous CAR T cells. There's a BB2121-7, which uses a PI3 kinase inhibitor to, again, look at to alter the constitution of the T cell population, and also a BCMA targeted CAR T cell that used a transposon-based technology to create the CAR T cells, both of which could significantly shorten the manufacturing time as well. Now, in addition, one of the other ways to get around the limitations of an autologous CAR T cell is to use a bispecific or a T cell engager. And there were a number of trials that were presented. The most interesting data came from the teclistamab that targets BCMA, as well as the talquetamab that targets the GPRC5D. Both these studies, with roughly about 100 to 150 patients, shows an overall response rate that's upward of 70% with fairly durable responses based on the follow-up that we have at this time. Some of the toxicities are similar across the bispecifics and the CAR T cells, like cytokine release syndrome, though the frequency and the severities can vary from product to product. We also saw some signal with neurological toxicity, especially with CAR T cells, and obviously that requires careful management, as does the risk of infection amongst these patients. Now, the use of, again, development of bispecifics that uses targets other than BCMA certainly enhances or expands the opportunities for use of this immunotherapy platform beyond BCMA. So the talquetamab targeting the GPRC5D, as well as the cevostamab, which targets the FCRH5, both of which appears to show some very interesting efficacy results. And we have to wait and see how the longer term data pans out. Finally, there's the ADC, the MEDI2228, which is a antibody drug conjugate, and that also shows significant activity in relapsed refractory disease, but has some issues in terms of eye toxicity that needs better definition at this point. So I think it's important, again, highlights the reason why we certainly want to consider putting patients on clinical trials, especially in the later stages of the disease, once they have exhausted the currently available opportunities or options, like the IMiDs, the proteasome inhibitors, and the monoclonal antibodies. In between, there's other drugs like the belantamab and the selinexor, both of which have been approved this year. And there was data looking at these two drugs in combination with the standard of care agents like pomalidomide, monoclonal antibodies, as well as proteasome inhibitors, showing that both these drugs can be combined with other standard of care agents to improve the outcomes of these patients. And then I think finally, obviously, we are all living in the era of the COVID pandemic, and we all have had to make significant changes with respect to how we manage patients with myeloma. There are a lot of guidelines that have already been published. I think we know that patients with myeloma tend to have a higher mortality, especially when they have uncontrolled disease. So I think the key message for patients with patients with myeloma in the era of COVID is that we should not compromise the care that they get in terms of myeloma control. Certain therapies can be delayed, especially in patients with the stable disease, but the primary goal should be to get the disease under control.